morning, our participants. We welcome all of you in the second day of our conference, the second international conference on molecular modeling and spectroscopy. We start this session today by session number 6, Keynote Lecture 2.
Dear Chairman, dear colleagues, our lecture is devoted to very important scientific and social problem. The coronavirus infection, COVID-19, caused by the SARS-CoV-2 virus, has suddenly swept the world. Due to the significance of the problem, the scientific world reacted with all its might. On the other hand, taking into account the need to urgently solve the problem of diagnosis this new disease using existing diagnostic platforms are being developed and put into production system for disease diagnosing. In parallel, with an unprecedented speed, there is an accumulation of fundamental knowledge about the SARS-CoV-2 virus. On this base, work is underway to create new immunochemical analytical platform for the diagnosis of viral disease. Given the constantly changing epidemiological situation, it is important that the platform created can be used to diagnose other dangerous and socially important diseases. In our report, we will consider some aspects of this multi-phased problem, primarily related to the new opportunities offered by the achievement of modern fundamental science. I will try step by step to formulate new opportunities and takes for the implementation of approaches that can open a new page in analytical chemistry of complex biological systems important for medicine. Selective reagents are particular importance in the analysis of various biopolymers. In this case, we will talk about selective bioreagents, which are most often the corresponding complementary nucleic acid and proteins. We will mainly talk about proteomic and analytical technologies, which are the basis for modern molecular diagnosis. Now, when we know the structure of proteins involved in the complex immunological processes at the atomic level, the analytics of biopolymers gains new opportunities and enter a new period of its development. Let's start consideration of the coronavirus system from the point of view of diseases molecular biomarkers. All diseases are accompanied by changes in the functioning of the body's biomolecular system and changes in the composition of the biopolymer compounds and metabolites. Therefore, a number of nucleic acids and proteins can be biomarkers of specific diseases. Hence, modern medical diagnosis is based on the determination of a set of biomarkers of diseases, which is directly related to capabilities of modern analytical chemistry. Consider the known biomarkers of the SARS-CoV-2 coronavirus. It are RNA, protein markers which including virus proteins, proteins of the immune response to infection, antibodies. A total of 27 COVID-19 proteins biomarkers were found. To date, the genome and protein of the SARS-CoV-2 coronavirus have been fully deciphered and shown as shown in this slide. Within the framework of this report, it is not possible to consider in detail all the available information on the functioning of the complex coronavirus system. Therefore, I will concisely demonstrate the information received in a short time, which allows us to generally understand the multifaceted nature of problem. We have detailed information about the life cycle of coronavirus reproduction and host cells and the trace that this process leads to molecular biomarkers of the disease. The picture of these processes is generally the same for many viral diseases as can be projected on coronavirus diseases. Finally, there are strict ideas about the ways of forming antibodies to coronavirus as an immune response to infection. All this makes it possible to identify the main groups of diseases biomarkers and the corresponding method for molecular diagnostics of the disease. PCR, polymerase chain reaction, a test for presence of the virus in the body. Test for antibodies and other protein biomarkers of disease, which occur during the body's immune response to infection. The key to creating molecular diagnostics is the identification of molecules in the body, the presence of which can be clear marks of diseases. The next slide demonstrates the main approach to the development of biomarkers of infection disease. The development of protein markers in infectious diseases include three stages represented by columns on this slide. The first stage provides for obtaining the initial genetic and proteomic data of the studied subject. As the second stage, all the data are analyzed and the most probable molecular biomarkers of diseases are identified. The third stage provides a deep interpretation of mechanisms of the imaging of specific biomarkers in the body during viral infection. I will focus only on the molecular proteomic diagnosis of disease. An important stage of the study is to research the dynamics of the accumulation of antigen antibodies in the course of the immune response to infection. This slide shows the dynamics of accumulation of immunoglobulin M antibodies and immunoglobulin G antibodies in coronavirus disease. It can be seen that immunoglobulin M antibodies appears in the first phase of the disease and only in the second phase of the disease. There is a sharp accumulation of immunoglobulin G antibodies, which provide long-term immunity. Modern science is not satisfied with the study of the problem in a qualitative descriptive level. It operates with molecular structures at the atomic level and specific interaction of biomolecules. Therefore, we will briefly dwell on the structure of the virus and the proteins structure of the coronavirus system. This slide shows the structure of the SARS-CoV-2 coronavirus and one of its most important proteins, a spike protein that recognizes the site of attack of host cell and triggers the mechanism for introduction the virus RNA into the cell and further reproduction of virus in the cell. Literally, in the first 2-3 months after the detection of new diseases, important information was obtained about the protein structures of the new coronavirus infection. This slide compares of three-dimensional structure of the SARS-CoV-2, SARS-CoV and MERS-CoV proteins. Taking into account the homology of these proteins and the direct measurements by integrating the previous known complex structures of SARS-CoV or MERS-CoV with antibodies, the complex structure of the spike protein of the SARS-CoV-2 virus was predicted. It is striking at what unprecedented speed were studies of structure of proteins of the coronavirus system with the resolution of the atomic level. In this regard, I would like to dwell on modern method for studying protein structure. The three-dimensional structure of proteins is determined namely by method X-ray structure analysis, MNR spectroscopy, cryogenic electron microscopy. X-ray structural analysis assumes the presence of a crystal. This method gives a resolution of up to 1 angstrom. But in the case of protein, the problem is that not all of them can be crystallized. NMR spectroscopy allows you to describe the structure of protein in solution. However, NMR spectroscopy only works well for relatively small protein molecules. Let us dwell on the younger method of cryo-electronic microscopy. Dobotic, Frank, and Henderson shared the 2070 Nobel Prize in Chemistry for developing this new method for obtaining 3D structures of biomolecules. The methodology of this method, until recently, seemed fantastic. An electron beam irradiates the frozen protein solution. The resulting scattering electrons create an image from which their structure can be determined. Cryogen electron microscopy is based on not only high-resolution electron microscopy, but also about all mathematical processing of set of images of variously oriented macromolecules. Subsequent improvement in electron microscopies and the software used to transform the images into three-dimensional structures made it possible to favor this technology over X-ray crystallography. The possibility of cryo-electron microscopic studies can be seen in the study of complex supramolecular complex of SARS-CoV-2 and RNA polymerase is RNA before and after RNA translocation. And so there is a more complete information about the 3D structure of the proteins of the SARS-CoV-2 coronavirus system. How can we use these new fields of fundamental science? How to use this invaluable information for the benefit of person? Now we have briefly considered the object of diagnostic research. We can move on the actual analytical problems of diagnostics. Determining protein biomass is a complex analytical task. The protein diagnostics, the studied biological object, contain extremely complex mixtures of proteins in a wide range of concentration. Thus, this task is often to determine very low concentration of biomass against the background of hundreds of thousands of other proteins. There are two main ways to solve the problem of proteomic analysis. First, purely instrumental approach is mass spectrometry with soft ionization method, tandem MS-MS analysis. The mass spectrometric approach requires extensive equipment. Second, the chemical method of diagnostic is the immunochemical analysis, to which this report is devoted. First, it is necessary to determine the place of immunochemical analysis among another methods of analysis. The following table is such attempt. There are 4 types of analytical definition. Immunochemical method of analysis belong to the first type of analytical process. It is remarkable feature of the separation of function of selectivity and selectivity between different chemical particles of the analytical process, namely between the bioreagent for diseases markers and its analytical level. This makes it possible to optimize this function on the analytical process separately to optimize the fine structure of the respective chemical particles. Thus, immunochemical analysis is a reagent method with the formation of a complex of an analyte with a conjugate of a bioreagent with an analytical label, and include the following 4 steps. The analytical process ends with the detection of the analyte complex with the conjugate visually or instrumentally. In this case, the selectivity and the cost of determination biomass is preliminarily determined by the structure of the protein, bioreagent, and the method of its production. Therefore, we will briefly dwell on the method of obtaining bioreagent for biomass of disease and the platform for analytical diagnostics corresponding to them. Method for obtaining bioreagent for biomass and the corresponding platform for analytical diagnostics can be classified in the form of a table presented on this slide. Most of the platforms in practice are based on the method using animal immunization. Method without extremely and time-consuming animal immunization are very attractive. They reduce the cost of the developing and manufacturing on wide range of highly specific biological components of test system. It is promised to use highly efficient technology of phage displays and target-specific aptamers, which makes it possible to obtain high specificity agent without the stage of animal immunization. We are on the cusp of the future proteomic analysis based on a molecular bioreagent design. There are 2 most productive ways to create proteins with new function. Experimental method of direct evolution that generate new proteins by simulating natural evaluatory processes. Second, computerial design of protein based on a combination of chemical and biological intuition, a known relationship between sequence, structure, and function of protein. The main backgrounds for successful molecular dynamic studies of protein interaction of components of coronavirus system for analytical and therapeutic purpose are availability of almost complete information about 3D structure of the protein of the SARS-CoV-2 coronavirus and a number of its antibodies. Second, high level of software for virtual molecular dynamic study of protein interaction. Third, availability of supercomputer for such research. The task is to optimize protein-protein interaction in the analyte bioreagent system by varying the structure of the bioreagent. There are two ways to use the information about the protein structure of coronavirus system. First, creation of new effective therapeutic agent for the treatment of coronavirus disease based on the deep understanding of protein-protein interaction. Second, creation of fundamentally new approach to the molecular diagnosis of coronavirus disease based on the analytical chemistry and the proteome. We will consider only the second direction from the standpoint of protein-protein interaction. The immunochemical analysis selectivity is provided by complementary affinity interaction of protein. This is clear manifested in the interaction of native antigens and antibodies. Can this interaction and selectivity be enhanced by creation artificial structure? Let us consider this easier using the example of protein structure of coronavirus system. The structure of the coronavirus spike and its receptor binding site RBD, due to which the virus binding to the surface of host cell has been established at the atomic level. This structure is shown on this slide. The spike protein receptor binding domain is an immunodominant and high specific target of an antibody to SARS-CoV-2. This slide shows the spike protein complex with the human ACE2 receptor from a different angle. The affinity binding of coronavirus spike protein to the ACE2 receptor can be clearly seen. This slide shows the result of structural modeling involving spike and ACE2 epitopes. At the same time, epitope region were identified for 8 antibodies of RDD, anti-RDD antibodies shown to neutralize active SARS-CoV-2. All this indicates the homology of proteins of various coronavirus system. Let us briefly dwell on the method of molecular dynamics in the study of protein-protein interaction in the coronavirus system. How can the possibility of computational design be realized based on the 3D structure of protein? The answer is to use the law of molecular dynamics to identify the behavior of proteins, to suppress the pathogenic biological function of or to provide high affinity interaction with analysis of proteomic diagnostic system. For this, it is necessary to carry out numerical molecular dynamic study of protein system at the atomic level. Why do we need a numerical experiment? Simulation. Simulation is a powerful tool that can replace experiment, simulate the setting of a new experiment, explain and give an understanding of the result of the experiment, compare theory and experiment. In this case, a cyclic path of cognition is realized. This slide shows the relationship between molecular mechanics and protein modeling. All this as a whole gives an understanding of the structure, dynamics, and biological function of molecule. The molecular dynamics method is to study the movement of atoms and molecules in time. Calculation of molecular dynamics make it possible to study the stability of both individual molecules and their complexes, and makes it possible to assess the significance of possible conformational rearrangement. Molecular modeling offers many possibilities. Using molecular dynamics modeling, it is possible to predict protein structure and complete viral particle models. Molecular dynamics methods are highly predictive. The application of molecular dynamics to protein design include following possibilities: designing stable protein variants, designing functional areas, providing insight into the unfolding folding pathway of the protein, construction of protein-protein complex and docking research, study of post-translation modification. The goal of molecular design is to optimize the structure of the bioanalyte and increase the affinity of interaction between proteins. This is achieved by increasing the area of intermolecular contact and the number of intermolecular bonds between proteins. After computer design of new structures, it is necessary to extremely study the interaction of this bioreagent with virus. A striking example of this approach is the work of Gorshkov et al. This work is devoted to the creation and use of SARS-CoV-2 pseudovariants conjugated with quantum dots on the surface of which there are spike proteins. These pseudovariants can mimic the interaction of the virus with the enzyme E2 receptor on the host cell surface and with antibody. The development of fundamentally new platform for diagnostics coronavirus disease requires extensive research in various fields of knowledge. What could be the role of analyst chemist in creating a new platform for diagnosing viral disease? Apparently, it is not realistic to expect from them a quick operation receipt of structural data of a new virus system at the atomic level. Fundamentally, for us, structural data obtained in the world's leading center became the property of all mankind. The task for chemists is to use this data for the numerical analysis and design of new effective protein bioreagent and their conjugates with analytical level. On this basis, new progressive analytical platform can be developed for the diagnosis of the viral disease. Conclusion. During the pandemic, diagnostic tools were developed hastily in fair order based on existing platform. It is necessary to look ahead and create new and more advanced platform for diagnostic of disease. The COVID-19 pandemic simulates the scientific community to unite and work toward a common goal through the interdisciplinary research. The ever-changing landscape, growing data set, and the need for quick action make computational research very important. It is important to develop new platform for diagnostics, taking into account fundamentally new knowledge at the level of molecular structure of virus and protein marks of disease. Another direction related to immune analysis is the creation of a new highly effect level for protein bioreagent, in particular fluorescent level with down and up conversion. It is important to obtain conjugates for protein and levels with an optimal binding site and relative orientation of protein and level. Thank you for attention.
Good morning. I am Paola Grenni from National Research Council Water Research Institute in Italy, in Rome. And first of all, I would like to thank Professor Medat and Professor Annan for their kind invitation at this prestigious conference. And I would like to also to thank all the people of the conference board and the organizing committee. And a special greeting to my friend Giuseppina Capriotti. My presentation is the study of natural microbial community as a tool for assessing the recovery of polluted environments. The National Research Council in Italy is the largest public research institute and the only one under the research ministry, performing multidisciplinary activities. And the main missions is to perform the research in its own institute to promote innovation and competitiveness of the national industrial system, to promote the internationalization of the national research system, to promote technologies and solutions to emerging public and private needs, and to advise government and other public bodies, and to contribute to qualification of human resources. As you can see in this map, the institute, many institutes of the National Research Council are distributed in all the Italian territory. Institute, so my institute in which I work, has 5 branches. The headquarters is in Rome, Montelibretti. There are other 2 branches in north of Italy and other 2 branches in the south of Italy. My specific group work in environmental microbiology, and we work in particular with bacteria and microbial communities. Why microbial communities are so important? Because microbes have several functions. In fact, despite their small size, natural microbial communities have a huge impact on our environment because they provide benefit to humans, the so-called ecosystem services. Ecosystem functions provided by microbes are the transformation of inorganic carbon into biomass by primary producer, nutrient regeneration and cycling. For example, microbial communities are involved in the carbon cycle, nitrogen cycle, and phosphorus cycle, and they transform the organic matter into other things of the food web into living biomass, regulation of biochemical cycle, and consequently climate. Community have a key role in removing xenobiotics. In this sense, they provide a very important function for the water and soil quality. In fact, several bacterial strains have been isolated in our institute for the capability to degrade herbicide, biocides, chlorinated solvents, PCBs, and etc. Functions such as degradation of organic matter, including xenobiotics such as pesticides, PCBs, etc., require complex metabolic pathways, and the recovery from contamination is only possible if toxicity of the contaminants does not hamper microbial activity, and knowledge of the natural remediation capacity of microbial community makes it possible to assess how much contaminants can enter higher levels in the ecosystem food web. Potential, microbial communities are able to break down of an enormous number of organic molecules, including xenobiotics, such as, for example, PCBs or pesticides, or other molecules that are called emerging contaminants, such as, for example, antibiotics and, in general, pharmaceuticals. In this slide, you can see that a chemical compound can be subject to photodegradation, evaporation, adsorption, sharper leaching, and a particular emphasis is the microbial degradation. The microbial degradation is the degradation of a chemical compound by the microbial community. In fact, as you can see on the right in the graph, in a sterilized soil, so without any bacteria or fungi, the degradation of a compound is very, very slow. On the other side, in the presence of a microbial community, so the non-sterilized soil, the degradation of, for example, an herbicide can be more quickly. So the presence of an abundant and varied microbial community is in any case a necessary prerequisite for an immediate and effective response to the various chemicals that can contaminate an ecosystem. On the left, you can see the principal diagram for degradation of a xenobiotic compound. So a xenobiotic can be degraded by the bichromate community or can be degraded by chemical degradation. And many metabolites can be formed, but only microbial community is able to completely degrade the compounds or the metabolites, so it is the only group that is able to mineralize the organic compounds to CO2 and water. On the right, you can see that in some cases, in fact, if a compound, if a xenobiotic is degraded, some metabolites can be formed, and if this metabolite can be more toxic than the main compound, and can be more persistent. So it is important to see not only the disappearance of a chemical compound, so a xenobiotic, but it is important to see also the degradation, a complete degradation of the compound and the metabolites. The water and soil quality is regulated by microbial communities. The ability of soil and water to recover from chemical contamination is primarily dependent on the presence of microbial community with the ability to degrade and consequently to remove the contaminants. So it is important to see and to study the microbial community. But the effect of a contaminant of a natural microbial community can be that of selecting specific bacterial population or bacterial strains able to remove the contaminant by using the chemical as a source of carbon or nitrogen. However, it can also happen that the contaminant causes the disappearance or the inhibition of some microbial groups, for example, those involved in nutrient cycles, in organic matter decomposition, and so on, with the consequence of loss of key ecosystem functions for maintaining water and soil quality. So the knowledge of ecosystem services provided by natural microbial communities is a crucial point for managing the recovery of contaminated water and soil. Although the potential homeostatic capability of microbial communities to remove contaminants from water ecosystem is enormous, it is not yet fully exploited. So the effect of contaminant on natural microbial community can be evaluated in terms of changes in microbial structure, for example, the dominance or the disappearance of some bacterial groups with consequent loss of an important ecosystem function, and the evaluation of the presence of specific bacterial population adapted to the contaminant and able to use it as a source of carbon. The study of microorganisms and the possibility of exploiting their natural recovery abilities is the basis of the strategies of the bioremediation, a treatment that promotes the ability of natural or selected microorganisms in the laboratory to degrade organic molecules. The biotransformation, for example, is the transformation into another less toxic or non-toxic molecule operated by microbial communities. Biodegradation is the breaking of the parental molecule into smaller or inorganic molecules. Mineralization is the complete biodegradation to inorganic constituent, such as CO2 and the bioremediation can be placed in situ, for example, as you can see here on the left, bioventing, biodegradation, biostimulation, bioaugmentation, natural attenuation, and phytoremediation. The last one, the phytoremediation, is the use of trees or other plants able to promote the degradation of contaminants in the soil, and in most of cases this degradation is operated by bacteria or fungi. So in this case plants promote the degradation operated by microorganisms. The bioremediation can be take place also ex situ, in which the contaminants or environmental samples containing contaminants are taken from the site and brought elsewhere for their treatment. Microorganisms play a key role in ecosystem for their homeostatic ability towards xenobiotic molecules. Removal of contaminants is possible to the extent that their toxic action does not prevent or inhibit their activity, and their natural recovery capabilities can be exploited for the reclamation of contaminated areas. The bioaugmentation, for example, is the addition of microorganisms which transform or biodegrade the contaminants. The biostimulation is the addition of in situ nutrients, oxygen, donors, electron acceptors, etc., in order to increase the population or the activity of naturally present microorganisms capable of degrading the contaminant. But the microbial ecology that study the bacteria, the fungi, so the natural microbial communities, depends on the methods capable of identifying and characterize structure and function of natural microbial communities. The morphological and physiological properties of the bacteria, for example, are not enough for classifying them from a physiological point of view, and most bacteria are not cultivable by classic methods. In fact, only less than 1% of the bacteria, of the natural bacteria present in the environment, are cultivable. The use of molecular methods makes it possible to overcome the identification limits and allow microbial phylogenetic identification, The latter is based on the sequencing of the gene codifying for the bacterial 16S rRNA or 18S for eukarya. At our lab in Rome, we used several methods. For example, the counts of microorganisms, the total counts, the cell variability, the characterization of microbial community structure with fluorescence in situ hybridization, and the ester liquid fatty acid to obtain a phenotypic fingerprints of the main microbial groups. Moreover, we perform the microbial community activity, in particular the dehydrogenase activity, the PCR and qPCR. The evaluation of the natural capability of recovery for contamination of polluted soil and waters at IRSA is performed by site-specific studies in microcosm, mesocosm, and then in on-site experiments. Here you can see a scheme in which you can see some microcosm with soil or water. Macrocosm and mesocosm are model ecosystems in which a portion of the natural environment is circumscribed and studied. They contain natural microbial communities that are maintained under controlled conditions such as temperature, light, oxygen, etc., but reflect the natural conditions. So, in this sense, if we treat, for example, some microcosm with a pesticide or with a xenobiotic, we can see we can study the microbial community, and we can analyze the degradation of the contaminant. In natural soil or water and treated with the contaminant are compared to microbiological control, so not treated macrocosm, in order to evaluate the effects on the natural microbial community of the xenobiotic, the degradation rates of the contaminants and the degrading partners, the presence of native microbial population that can metabolically or co-metabolically transform organic contaminants, and in some cases isolate and identify microbial strains capable to degrade the contaminants and usable in bioremediation interventions. In this slide, you can see the methods that we used that I told you before. So, we analyzed the microbial abundance, the cell viability, the fluorescence in situ hybridization, the dehydrogenase activity, the NGS, PCR, qPCR. We applied these methods to several studies in order to assess the effects of selected contaminants on microbial communities of different ecosystems. For example, herbicides and pharmaceuticals are used in large quantities and consequently can cause diffuse or point contamination of soil and water ecosystems because of their chemical characteristics and persistence. We performed some studies regarding pesticides, in particular in soil and in groundwater. We perform also experiments regarding pharmaceuticals with different pharmaceuticals, for example, the antiviral Tamiflu, some antibiotics, and some experiments regarding PCBs and heavy metals in an area contaminated by heavy metals and PCBs, and in water. In this case, we assess the absorption of heavy metals by microspheres. In this presentation, I would like to present some studies regarding the antibiotics and PCB and heavy. As you can see in this graph, the pharmaceuticals, and in particular the antibiotics, can be entered in the environment. In fact, antibiotics that are considered emerging contaminants because they are not assessed in different soil or water environments are only partially metabolized in treated organisms and through exertion reach wastewater treatment plants or soil. Most of the water treatment plants are not able to remove the pharmaceuticals efficiently, and through their effluents, they reach surface water. The use of savage sludge, such as biosolids, and manure as a crop fertilizer and irrigation with reclaimed water will entrain human and veterinary antibiotics into agricultural soil. A transfer of antibiotic from wastewater or animal manure through soil to edible crops is also possible. You can see an example of a degradation of some antibiotics that we performed in microcosms, in which water of the river water was treated with these contaminants. So we evaluate the effects of a fluoroquinolone and a sulfanilamide on the natural microbial community of the river Tiber. In particular, we assess the disappearance of sensitive populations involved in key ecosystem services, the increase in bacteria-resistant populations and their relative genes, and the promotion of degradative populations. In this case, ciprofloxacin was quite persistent, and sulfamethoxazole was biodegradable. Ciprofloxacin had a long-term effects on gram-negative bacteria, and sulfamethoxazole promoted the growth of some bacterial groups, and the biodegradation was a key mechanism to remove these antibiotics. In this slide, you can see some papers regarding the antibiotics and our experiment that we performed in our lab. The other example that I would like to show you is regarding an area in the south of Italy in which the soil is contaminated by PCB and heavy metals. In this case, we performed the photo-assisted bioremediation. The area is in the south of Italy near Taranto, and in this case we planted several popular cuttings of the hybrid popular genotype Monviso, and this plant was planted in 2013. and since that date we follow the PCB degradation and the heavy metal disappearance from the soil. In fact, a preliminary chemical analysis showed that there was a diffuse contamination and non-homogeneous contamination of PCBs. In this case, we use the phytotechnology for the soil cover. In fact, plant-mediated decontamination and detoxification processes. For example, the success of fetal remediation is dependent on the potential of the plants to yield high biomass and withstand the metal stress and the metal bioavailability in rhizosphere soil. Microorganisms, in this case, can have a key role in metal removal because they are able to enhance their translocation or to reduce their mobility availability in the rhizosphere. In the case of PCB, microorganisms are able to degrade this very persistent contaminant. These are only few pictures of the area in which we planted the populus. After 40 months, as you can see, the populus growth. And after 2.5 years, the populus is very high. From the rhizosphere and from the bulk soil were sampled at different times, for example, after some months or after 2.5 years. We found at day 0 PCB contamination together with the presence of heavy metals in the subarea sampled. However, PCB and heavy metals concentration decreased below the Italian legal limits 1 year from the popular plantation. Assisted photoremediation performed at the Taranto study area. The comparison of the initial chemical and microbiological analysis with those performed after 2.5 years the popular plantations shows that plant microbial interactions played a key role in promoting heavy metal remediation. The significant reduction in heavy metals showed an overall positive effect on the bacterial community. The highest number of bacteria detected were in line with the lowest metal concentration, and inside some bacterial groups, such as alpha, gamma, and firmicutes, several bacteria able to resist or to remove heavy metals in different ways are reported. Popular assisted bioremediation, owing to its economical, environmental friendly, and landscape enhancing, is a green remediation, strongly advisable in recovering areas of diffuse and heterogeneous contamination. In this slide, you can see some publication regarding the area, the Taranto area. Now I would like to show you some results regarding the project entitled Spectroscopic Analysis of Natural Microsphere for Maximizing its Application in the Remediation of Inorganic Pollutants, that was performed in the frame of a cooperative program of the Agreement on Scientific Cooperation between Italian National Research Council and the Egyptian National Research Center. This project was performed with Medhat Ibrahim, Professor Medhat, and gives the possibility to have several exchanges between Italy and Egypt, which has the potential to remediate aquatic environments from pollutants. And the microsphere prepared with hyacinth and chitosan from cross-linked sodium alginate and water hyacinth root were performed at Egyptian National Research Center. River water microcosm containing microsphere and treated with heavy metals were performed at Water Research Institute. In order to evaluate the effectiveness of microspheres with water hyacinth to absorb metals, in particular cadmium and lead. Evaluate the effect of macrosphere in the presence-absence of the metals on the river natural microbial community. You can see in this photo the microcosms that we performed at Water Research Institute, in which we had control, so only river water treated with metals, and river water treated with metals in the presence of microspheres, and river water with microspheres. The overall results show that the microspheres were able to remove high heavy metals, about 1 mg per liter, from river water in a short time, and the microsphere had a positive effect on the natural microbial community of the river water, probably through the release of organic carbon. The microsphere were able to keep metals adsorbed for a long time. The results show that the green technology proposed is an effective remediation method for lead and cadmium removal from river water, and is a very promising, not least because of its low cost. Some shortcomings and challenges, and the conclusions. Biodegradation is a key mechanism to remove organic contaminants from the environment. Thin information gaps on the ecosystem services provided by the natural mitigation capability of microbial communities is a challenge. Develop bioremediation techniques from environmental purposes through the isolation and utilization of specialized degradative microbial population could be very, very useful. And combine the information obtained from the chemical and physiological data together with the microbiological ones is very useful. Thank you for your attention, and thank you again to our Egyptian friends, and I hope to see you very soon in Italy to exchange another time our experience.
Thanks a lot for this invitation. Our today talk about recent progress in ultra-fast lasers and laser spectroscopy and its environmental applications. Outlines of this talk about ultra-fast lasers and generation fight light sources, exploring the ultra-fast dynamics and switching molecules and its applications, LEAPS, spectroscopy as analytical technique, environmental applications, then conclusion. Building and ultra-fast lasers. How to build white light radiation source of ultra-fast laser pulses. Our schematic diagram describes our system composed of main laser source of 800 nanometer, 32 femtosecond, 2.5 millijoule maximum energy, using some kind of optics which focus this laser into hollow fiber tube filled with some inert gas. This kind of fiber help the laser to generate a supercontinuum emission. After this, we have a kind of suitable mirrors and sharp mirrors for compressing the pulses and getting something like from 4 to 7 femtosecond pulse duration with a high energy. This is the optical layout of the few cycle laser system, which contain the seeded oscillator as a script and CPA sharp pulse amplification system, which pump the high power laser to the hollow fiber compressor. Then we have spider and CCD camera for measurements the laser characterization. The first publication about this system was presented in the conference ATO 3 in 2011 during the presence of Paul Corkum, the father of ATO second system. The system has been built in King Saud University in semi-clean room, and the output has been applied in many applications. This is well-known autocoloration intensity system at which one pulse is split to two arms. One of them is delayed using some optics. Then both of the arms are interfered using nonlinear crystals, at which intensity function of autocoloration can be given as a function of time. This is the temporal profile of the output of the system, which give pulse duration about 8.9 femtosecond. Then a kind of optimization has been done using different pressures for the hollow fiber tube, at which we reach something like 100 gigawatts at different values of pressures for of gas pressures. A full details about this system has been published in a book with the title Generation of Few Cycle Femtosecond Pulses via Supercontinuum in Gas-Filled Hollow Core Fiber. Then, as advanced enhancement of the output, we would try to enhance the pulse duration using a nonlinear kind of optics and increase the pressure inside the hollow fiber tube and make a relationship between the bandwidth and the pressure. Then we got a squeezed setup like this, at which we minimize the self-focusing problems, and we got an output up to 4.8 femtosecond after compression. Then we switch it to application of ultra-fast lasers in many areas. One of the most important common system at which we used our ultra-fast pulses is bump probe technique, at which we characterize some important ultra-fast dynamics and ultra-fast transitions, at which we can follow up the dynamics of this system using two pulses. One of them are bump pulse, which is slow pulse, about 30 femtosecond. The other pulse is fast pulse, is 5 femtosecond. The bump pulse excites the system to intermediate state. Then the probe pulse excites the intermediate state to another excited states, at which we diagnostic these ultra fast dynamics, like this one. As an application of ultra fast dynamics, we have developed a special kind of polymers called diraethylene derivatives, DMV, and using an ultra fast lasers we can control the structure of this special polymer from open to closed structure. The structure change due to an absorption of special photons, if the photons is UV or if the photon is visible. If the photon is UV, the structure will be closed, and if the photon is visible, the structure will be open. And transitions from open to closed structures, we have studied the ultra-fast dynamics and the transit transitions between these structures. And this is simulation demonstration of how the photons can change the structure of this special polymer. We have to take into consideration that these pulses is ultra fast pulses, and the structure is changes. From this experiment, we have concluded that chemical bond rearrangement during the photo transformation includes electronic as well as geometrical structures, changes the molecules. The molecular structure changes can be applied to various photonic devices. The electronic structure changes can be applied to optical memory media and various photo switching devices or molecular mechanics. As example for this, we can replace the integrated circuits, or mainly the transistors, with such kind of photo switch molecule, so that instead of making on and off with a semiconductor, we can make on and off 1 and 0 with photo switch molecule using this kind of ultra fast molecule, so that we can make the computer more faster than the usual one. Now we will transfer to LIBS, laser-induced breakdown spectroscopy, as elemental analytical technique. Laser-induced breakdown spectroscopy is a type of atomic emission spectroscopy that uses a laser to vaporize or ablate a microscopic layer of sample's surface. The resultant plasma caused by this laser ablation process emits light as its cause. This is then followed by collection of the light and then analyzing it with a spectrometer for quantitative and qualitative material analysis. Now these are the lists of mechanisms which can be involved in laser interaction, depending on the type of the sample itself. For example, reflection can be involved in solid and liquid, but not in gas. Absorption is valid for solid, gas, and liquid. Melting is valid for solids, vaporization for solids and liquid. Plasma formation is valid for solids, gas, and liquid. Laser plasma interaction is valid for solid, gas, and liquid. Shockwave formation is valid for gas, solid, and liquid. Hydrodynamic expansion is just valid for gas. Ejection of solid target is just valid for solids. Summarize the interaction dynamics of laser with solid target. We start from A with some reflection and absorption of part of the laser beam, then after absorption heating up, melting and shockwave generation, then plasma formation and cooling down of the plasma, some ejection of solid targets and cooling down of the rest of the plasma. Why ellipse? Why we need to apply ellipse? Because it is atomic emission spectroscopy provides analysis of every element which can be allowed for quantitative measurements, real time with no sample preparation, and can be applied for solid, liquid, and gas and aerosol species, cheaper than other analytical techniques and need no any kind of aggressive chemicals. There are many parameters which may affect ellipse, like laser energy and radiation, pulse duration, laser wavelengths, ambient conditions like pressure and composition, matrix of the sample, thermal properties of the sample, focusing conditions. Of course, there is no complete models for to gather all of these parameters together, so that we check one by one to see the effect of these parameters on behavior of ellipse. Basic theory. Simply, we focus a high energy laser beam on an unknown sample to be analyzed. Then the generated plasma emission can be analyzed using a high resolution spectrometer, and the intensities of these emission lines can be identified for each element of the sample. Plasma and its parameters. Two main parameters can characterize the generated plasma: plasma temperature and plasma electron density. Plasma temperature can be estimated using Boltzmann distribution. From Boltzmann distribution, we can relate the intensity of the emission lines to the energy of the excited states and the generated electron plasma temperature. Moreover, this Boltzmann plot can give us linear relationship with the slope representing the temperature of the electron temperature. To calculate the plasma density, we use the stark broadening of the emission lines divided by the theoretically calculated stark broadening. An example of Boltzmann plot. Here we can analyze the composition of golden alloy using Boltzmann plot to estimate the temperature. The temperature in this linear equation representing the slope of the equation. Keep in mind that not any ionization can be considered to be breakdown of the sample. There is a threshold for this estimated to be number density of electron to number density of atoms equal 10 to minus 3. Presenting the plasma emission evolution in time domain. After few nanoseconds of plasma emission, continuum starts to come up. After about hundreds of nanoseconds, ionic emission lines can be expected. Atomic emission lines can obtain around 1 microseconds to 2 microseconds. Above 2 microseconds, molecular lines emissions can be observed. So measurements of plasma electron temperature using Boltzmann plots can be obtained using the proper gate time and late time for the emission lines of atomic, molecular, or ionic emissions, depending on the gate chosen by the detector. Also, by measuring the width of the emission lines, we can calculate the plasma density. One of the most important applications of LIPS technique can be shown here by analysis the soil using laser-induced breakdown spectroscopy. We collect soil from different areas in Sahara Desert and collect all of the samples in different locations and analyze these soil samples. The collected samples are pressed as a tablet, then examined to analyze the composition of these unknown samples by applying LIPS technique. By focusing high energy pulsed laser beam on the surface of the sample, and the emission of the generated plasma are collected using some fiber optics and lenses to be analyzed using high resolution spectrometer and CCD camera. The emission spectrum of the unknown lines can be obtained as explained before like this. Then the emission lines can be analyzed and identify the emission of each line, which is characteristic lines using reference source like NIST. By comparing the emission lines and NIST data, we can identify the emission lines and identify the composition of these unknown samples. This is an example of LIPS spectrum of soil sample in the north of the examined area, and the spectrum shows some kind of emission lines of calcium, magnesium, aluminum, and iron, silicon, and sodium, and so on. This is Boltzmann plot for the collected soil samples, which give us is an estimation of the electron temperature of about 8378 Kelvin, which can be calculated from the slope of this curve. If we have different concentrations from the same element, we can make calibration curve. This is a calibration curve for iron at line 404.5 nanometer at different concentrations and different intensities for the same line. This is calibration curve for magnesium and different concentrations of magnesium from 200 to 1200 ppm. This is calibration curve for sodium at line 588.9 nanometer from concentration of around 440 to around 600 ppm. This is summarize the observed detection limits of the observed elements available in the examined soil. We have observed detection limit of 2.6 ppm for magnesium and 1.6 for sodium. This is example of published papers and conferences for using LIPS in analysis soil samples. Now we can give a summary about other applications which can be involved in LIPS technique. We can use LIPS technique in industrial applications, for example, analysis of nanostructure titanium. LIPS limit of detection of elements in steel and aluminum alloys. Then we have improved detection limits of LIPS. We have published papers about fast nanostructure zinc oxides. In medical applications, we have published papers about quantitative analysis of dental nanocomposites. In environmental applications, we have published papers about analysis of seawater and calibration-free identification of seawater salinity, and analysis of agriculture drainage water, and discrimination of bacteria using LIPS technique. Developing of LIPS in liquids also has been published. One of the most important medical applications just published in cooperation with Professor Aslam Beg in Pakistan, Qaid Azim University, we have performed LIPS technique for kind of light. This technique in Pakistan to detect toxicity in some oral anti-diabetic drugs using LIPS and laser ablation time of flight mass spectrometry. In these experiments, we have examined a kind of counterfeit medication which may contain toxic elements with higher concentrations. We have applied LIPS technique and laser ablation time of flight mass spectrometry. We have made a cross-section in these kind of tablets, and we found that some heavy elements as a coating layer of about 500 micrometer of aluminum and other type of drugs we found instead of aluminum titanium with a very thick layer of 500 micrometer. The coating layers and the inner layers of the tablet, and we found many elements in these tablets like titanium and aluminum and calcium in different concentrations. We have found that the outer shell layers of these counterfeit medications composed of aluminum and titanium, which represent about 600 milligrams of titanium and 100 milligrams of aluminum, which exceed the maximum allowed concentration in the human body. In this manner, it is considered a very toxic drug. And we have published this paper recently in Journal of Microchemical. Some points as a conclusion: advanced ultra-fast femtosecond system, which has the ability to generate broadband spectrum white light with pulse energy of 0.6 millijoule and tunable pulse duration from 6 to 13 femtoseconds is demonstrated. The observed pulse has been compressed up to 4.8 femtoseconds. Using ultra-fast pulses, the dynamics of the photo-switching molecule has been explored. LIPS spectroscopy has been applied to analyze soil content and estimate the contamination concentrations. LIPS has been applied to detect toxicity in counterfeit medication. The observed ultra-fast pulses can be used to create more shorter pulses in the attosecond regime with shorter wavelengths in the UV X-ray regime in future. At the end, I have to thank all the international collaboration group, Professor Rick Turbino from Georgia, United States, Professor Takeuchi Kobayashi, Tokyo University, Japan, Professor Mustafa Sayed, Georgia Institute of Technology, USA, Professor Dong Kim, Bostik, South Korea, Professor Aslan Bek, Pakistan, Professor Fernz Krauss, Max Planck Institute of Optics, Germany, Professor Singh, United States, and thank you for thanks for all of you.
Good day, ladies and gentlemen. First of all, I would wish to thank to the organizer of this, the second online conference on molecular modeling and spectroscopy. Secondly, I regret I can't join my Egyptian friends, but to whom I wish a full success. And to all of you, Salam Alaikum. My presentation entitled Concordant Electron Paramagnetic Resonance and Thermoluminescence Ages of a Romanian Plain Loess Deposit is dedicated to a very actual problem of archaeology and quaternary geology. I mean, the absolute age of sedimentary formation no older than 500,000 years. Sedimentary material itself has the age of the parent rocks and not the age of the depositional process. This makes an age method based on the disintegration of the radioactive elements existing in sedimentary materials useless. This disadvantage can be overcome by using the accumulation of radiation centers produced by the natural radioactive elements existing in sediments. The absolute majority of sedimentary material contains the same average amount of natural radionuclides closer to the upper continental crust. I mean potassium-40, thorium-232, and two isotopes of uranium, 235 and 238. The alpha, beta, and gamma rays emitted by these elements generates at microscopic level dose rate which varies between 5 and 20 milligray per year. It is worth remarking that the mean lifetime of these radionuclides varies between 0.7 and 14 gigayears, so that their presence maintains for long period of time a constant level of internal dose rate. In this way, the natural radioactive elements generate in the composing minerals of sediments, in our case the loess, defects, structural defects, mainly consisting of free electrons which are trapped by the impurity levels existing in the forbidden band. Hence, the trapped electrons become excellent proxies for the sedimentary age, as at temperature above 500 degrees centigrade or at solar light, they recombine within the valence band with holes, and in this way they reset the age clock. If the environmental conditions remain constant for a long time, which is the case of loess deposits, the free radicals accumulate with a constant rate and their number increase monotonously with the age. In these conditions, the free radicals' annual accumulation rate is positively correlated with the content of natural radionuclides, whose content can be radiometrically evaluated. At its turn, the content of radiation-trapped electrons, radiation-free radicals can be determined with enough accuracy by electron paramagnetic resonance, thermoluminescence, and optical stimulated luminescence. In these conditions, the sample age finally can be calculated by dividing the free radicals number to the annual accumulation rate. Although intensively used in archaeology and quaternary geology, these methods have some drawbacks due to the long time stability of trapped electrons or to the soluble uranium carbonate circulation. Therefore, electron paramagnetic resonance geochronology has a precision of about 10%, recommending EPR dating to be checked by an alternative methods. In this regard, we have determined the age of a loess deposits from Gorgana first archaeological site in Romanian plate, Radovana village, by both EPR and TL, checking at which extent the age provided by those two methods are coincident within experimental uncertainties. As it is known, loess is an aeolian sediment consisting of fine grains of quartz, calcite, feldspars, smectite, chlorite, and other ill minerals, and mica group from a diversity of source lithologies. Loess deposits in Romania are covering mainly the southern and southeastern part of the country along the Danube River, where they form deposits up to 50 meters thick. These deposits contain several well-developed paleosol sequences, which represents an archive of the environmental changes in the past 800,000 years. Here you can see the map of Romania with the main loess deposits. As you can see, they occupy the southeastern part of Romania, especially in the vicinity of Bucharest and near the Black Sea. Here, not far from Bucharest, lies the Radovanu site, which age we have tried to evaluate by both methods, thermoluminescence and electron paramagnetic resonance. On this map, we have represented the position of Radovanu village with Gorgana first archaeological site, as well as a sequence of loess and paleosoil which we have investigated. This Gorgana site contains a fortress with earth walls, some of them being recycled during the Roman occupation. It happened in the 2nd century of our time. The yellow-browny soil, rich in loess, is characteristic for the eastern Romanian plain. For our study, we have chosen a level situated 2.8 meters below the surface. We have collected about 2 kilograms of raw materials, which has been split in 3 aliquots for further radiometric, electron paramagnetic resonance, and thermoluminescence. As free radicals are generated under the action of natural radionuclides, their content should be precisely determined. In the present case, this task has been accomplished by gamma-ray spectroscopy. Among different minerals fractions, the quartz one proved to be the most suitable for both EPR and TL determinations. Accordingly, the silicates were separated from the carbonates and organic fraction by dissolving the soil sample in a solution of hydrochloric acid, oxygenated water, and distillated water. After complete dissolution of carbonates, the solution was filtered and solid fraction with size between 63 and 90 micrometers was selected by sieving throughout a 170 to 270 mesh sieve. Further, by using a solution of sodium polytungstate, it was possible to select a mixture of quartz and plagioclase, the last one being removed by immersion in a solution of 40% hydrofluoric acid. This treatment removed also a superficial layer of quartz grains containing the free radicals generated by the external alpha rays. To avoid any error induced by bleaching, beginning with the extraction of soil from cores and finishing with EPR and TL measurements, all operations were performed under protective red light. Radiometric determinations were performed by high-resolution gamma-ray spectrometry, Canberra high-purity germanium detector with 40% efficiency and an epoxy windows of 0.6 mm able to register gamma rays energies lower than 40 keV. At the same time, the resolution of the spectrometer was about 1.96 keV for the 1033 keV line of 16 cobalt. The background contribution was reduced by placing the detector in a 747 Canberra lead shield covered with thin layers of ancient lead. At the same time, the contribution of background has been evaluated by measuring the counting rate for the entire spectrum for 3 times 33,000 seconds. We have checked also the existence of radioactive equilibrium for uranium and thorium radioactive series, and within experimental errors, we have found no differences in the specific activity of bismuth and lead for both radioactive series. Further, starting from the content of natural radioactive nuclides determined as mentioned before, the annual dose rates were calculated by using the existing content to dose rate coefficients. In this way, the final annual dose rate was determined with a total uncertainty no greater than 6.1%. It is worth mentioning that the precision and accuracy of radiometric measurements play a huge role in both EPR and TL absolute age determinations. The initial dose absorbed by investigating loss layer since its deposition and present time, I mean the paleo dose, was determined by the additive method. This procedure implied the irradiation of quartz fraction with cobalt-60 gamma ray at increased dose from 0 to 60 gray, all samples being placed to attain the electronic equilibrium in 1.5 mm perspex tubes. Irradiation were performed at room temperature using a gamma chamber at a constant dose rate of 6,210 gray per hour, monitored by alanine dosimeters with a total uncertainty of about 5%. All EPR measurements were carried at low temperature by using an X-band spectrometer provided with a transversal electric 102 rectangular resonant cavity and 100 kHz modulation. A polycrystalline sample of calcium oxide impurified with manganese 2+ was permanently kept in the resonant cavity as an external reference sample. To record the free radical spectrum, which has a J factor equivalent to free electron, we have used a magnetic sweep restrained to the space between the third and the fourth line of hyperfine structure of manganese 2+ ions, as the next slide illustrates. This slide illustrates first of all the shape of the EPR line of free radicals, amplitude increase with the absorbed dose. In the inset, you see the position of resonance line of free radicals with respect to the manganese 2+ hyperfine structure lines, which has been used all the time as internal reference. At their turn, thermoluminescence measurements were performed in the domain of 50 to 410 degrees centigrade with a heating rate of about 4 degrees Celsius per second. A monolayer of quartz grains was spread on a 5 mm diameter stainless steel, and thermoluminescence radiation was recorded by using a blue transmitting filter. Resulting TL signals has a characteristic glow curve with a maximum centered about 310 degrees, as illustrated in the next slide. Here you can see the thermoluminescence signal whose intensity increases with the absorbed dose. The final value of local annual dose rate in soil, as radiometric determined, was of 3.43 plus minus 0.21 milligray per year. Using the reconstructive dose technique, the paleo dose was calculated from a linear dose dependency of the EPR or thermoluminescence signals according to the following equation. It's a simple linear equation where the first term represents how it increased with the dose the amplitude of both EPR and thermoluminescence signal. Here, in this representation, both B-plots shows how the paleo dose can be determined by using the additive dose methods. Red points represent the amplitude of the signal after the samples were irradiated in laboratory, while the black dot, the black diamond, shows the initial amplitude of the signals without any radiation. As you can see, those 3 points plus the experimental one determine a curve, a straight line, whose intercept represents the paleodose. In this case, we have obtained very close value of paleodose, which means that both methods are confident for our measurements. Once the paleodose determined for both type of methods, the age can be very easily calculated by dividing the paleodose to the annual dose rates, following a simple mathematical relation used on the bottom of the slide. The final results concerning the age of loss layer gave results which are coincident within one experimental combined uncertainty. I mean about 20 kiloyears, which situate the site at the beginning of the first millennium, which coincides with the Roman occupation of this territory. As a final conclusion of this study, it should be remarked that in both cases the ages are affected by relatively high uncertainties of the order of 10 to 15%. They are significantly higher than in the case of other absolute radiometric methods such as potassium-argon, argon-argon, uranium-lead, all of them based on the disintegration of long-life natural radionuclides. But in spite of this, both methods we have used, although based on different physical principles, gave very close results, which recommend the conjoint use of EPR and thermoluminescence methods as an appropriate procedure for short and medium time ages, in our case up to tenths of kiloyears. At the end of this presentation, I would express our gratitude to organizer of this conference as well as to the honorary audience for their attention. Once more, thank you, all of them.
Assalamu alaikum and greetings from the Ohio State University. I'd like to thank Professor Medhat for inviting me to this conference. So today I'll be talking about spectra of phosphorus for astrophysical modeling. Phosphorus is very important at this time for the clue of extraterrestrial life particularly. So I'll be discussing all those and the spectra we have made, and I will also just name the people who also work with me are Bilal Shafiq, Michael Rothman, and Rala Nagma. So this week's, okay, or the last week's, the hot news was the detection of phosphine in the atmosphere of Venus. Venus is a twin, evil twin of the Earth because it has same size, about this, but it has a very hostile environment for any living form because the surface has temperature 900 degrees and the density is 90 times more dense than that of on the Earth. And it has carbon dioxide and sulfuric acid, but it has also cool air on the top, which has high speed going around it. So recently, the two detector, INCT and ALMA, detected phosphine in the atmosphere, and phosphine is produced by living forms, animals. And this is a big news because we have been searching for a life form outside the Earth, and this seemed like a potential clue to see something living outside our Earth. But there was a conclusion of this observation in the Nature letter written by Grievous et al. That predicted maximum photochemical production of phosphine, which is pH3, found to be insufficient to explain observations by more than 4 orders of magnitude. So the discrepancy, huge discrepancy between the observation and the prediction is the underlying science which was not treated properly. So today I'll be talking about the underlying science that we can do to explain the spectra. Okay, so we have been trying to find out any extraterrestrial life out there. We are just one star in our galaxy and there are millions of galaxies. And if we are living in our stars and it is found that all the scattered elements in the space are inside us, so that means we are made from star dust. That we have found that this formation of living form is something extraterrestrial process. So it is expected or indicated that there will be life forms out there. So we cannot be just unique in one star. So this systematic study started in 1984 with this program called Searching for Extraterrestrial Life, or SETI. It is based in California, and it has been trying to get any kind of signal which has meaningful signal to find any life form, and it has not detected any so far, except it got some kind of signals, particularly the one in 2004. There could be something life form, but they are not analyzed yet. But it has been searching through 20,000 red dwarf stars, which usually hold exoplanets, and to find any sign of life form, and it expects to say some confirmation in 2040. Okay, this interest has gone up considerably since the detection of exoplanets. The sun is our star, and we have 8 planets. We call them planets. But if any other star which holds a planet, we call that planet as exoplanet. So even 30 years ago, we did not see any exoplanet. Even though we had speculation that exoplanets exist. Now, in 2004, we had a direct measure of this star far away from us with a planet going around it. So this was the direct first clear image of an exoplanet. And until now, we have detected over 4,000 exoplanets in our galaxy. And those are largely detected by this Kepler observatory. And there are also other ones in the HARPS and Hubble Telescope and Spitzer, Kehl, TRAPPIST. They have also been detecting a lot of exoplanets. It is important to know the exoplanets because ultimately we have to find an habitable planet where the human race can move to. The biggest one that is going to space next year is the James Webb Space Telescope, or JWST telescope, which is a huge one, which is 18 meters compared to 2 in Hubble telescope and mass of 6,500 kilograms. But the one main objective of this JWST will be characterize exoplanets and see what molecules their atmosphere contain. So it will be detecting photons in the infrared region from 6 to 28.5 micron. So if we look at the atmosphere of our planet, we see all kinds of biosignature elements. Biosignature elements are like water, of course, water and carbon dioxide, methane, nitrous oxide, ammonia, etc. But there are also basic elements of life, which are carbon, nitrogen, oxygen, potassium, calcium, iron, and also phosphorus. Even though in our atmosphere you can see all these molecules which can be detected from outside, but are there also basic elements living here? So you can see this is the picture of a DNA, which is other carbon, nitrogen, oxygen, but all the red dots, they are like phosphorus. So phosphorus is a basic element of life. Okay, but what happened, even though in space we find our body elements are scattered in space, but we don't see phosphorus. Strangely, this is a picture that cartoon published by New York Times in 2015, showing that cosmic selves, which has all the elements of the space forming, living for us. But only the question was, why can't we detect phosphorus compared to all these elements that we are seeing? So this brings the question, is phosphorus is a clue for finding extraterrestrial life? Okay, but we know that phosphorus forms during supernova explosion, when a very massive star just collapses after burning out all its hydrogen. That extreme pressure and temperature forms these heavy elements and is scattered in the space. But we know that Cassiopeia A, this supernova remnant, also produced phosphorus. But we find that there are other supernova explosions which did not produce phosphorus. And luckily, it seems our sun has abundance of phosphorus. Okay, so there's this a survey, All Sky Survey, STSS, which has this project called Apogee to find out the biogenic elements in space. You can see there's phosphorus, which is red, is in our body, and they try to detect phosphorus and all these signals coming from detected from various astronomical objects, but there are not much phosphorus except tiny bit here. So phosphorus has not been detected. But even though it is not detected, we can predict where to look for, and if we see something, how to analyze them. So how should we do it? It is through spectroscopy. So how much do you know about phosphorus? Phosphorus is abundant on Earth. We have been using it for our application industries, fertilizers, etc. And we know it glows it in the air, but it is also highly reactive. Since it's not seen in the astrophysical spectra that much, phosphorus is one element which has not been studied extensively. So we have very limited knowledge about phosphorus, and that's what we will be talking about, how much we have learned. So one atomic process that we studied for some time, it is about excitation and de-excitation, like electron going from one excited state to a different excited state by absorbing a photon and then coming down by giving out the photon. So we can have absorption lines, also emission lines in the spectrum. So we have studied many transitions of phosphorus, not only one ion, starting phosphorus 1, which is neutral phosphorus, going up to phosphorus 15. Phosphorus has 15 electrons, so that will be like hydrogen-like phosphorus. So we have calculated all transitions, many transitions compared to whatever is available, and we formed a spectrum. And you can see the spectrum over here. I'm just presenting only 5 spectrum instead of 15. The top one showed phosphorus 1, 2, 3, and the bottom one, the last ionization stages of phosphorus, phosphorus 14 and 15. And we can see that in our regions of lines from X-ray to far infrared. So this one, which is highly charged phosphorus, we expect that it will be seen in the X-ray region. This is X-ray. Only in the X-ray region we can see phosphorus 15. 14 can be seen in the ultraviolet regions too. Then phosphorus 1 is, I can see, it can be seen in the far infrared region. Phosphorus 2 seems to be visible in the visible region quite a bit, and phosphorus 3 is in again in the ultraviolet region. So we plan to plot them and analyze those where this particular lines could be strong, which can be searched in the astrophysical spectra to find the clue of phosphorus existence. So this is what we have done, and how accurate are our numbers. So this shows a table of comparison of A values or radiative decay rates. These are our rates and these are the existing values. I just picked some limited transition for each ion, phosphorus 1, 2, 3, 4. I could not go all the way 15 because of the size of the page, but comparison shows that our values agrees with the best values or whatever available from very good to a reasonable degree. So that shows the accuracy. We plan to do more accurate calculations using our matrix method later on. So this is our predicted spectrum, but this is not only one way to analyze the phosphorus, because phosphorus lines can be produced by 3 other atomic processes. One is called photoionization, like the ion is here and photons are traveling. One electron can absorb the photon and leave the ion. So this is ionization, and the process called photoionization. So it can happen directly that the ion absorbs the photon and get ionized. We call it like direct photoionization. But it can also happen that between the initial and the final state, one intermediate state forms where 2 electrons are excited. We call it doubly excited auto-ionizing state. This happens all the time in the nature because these are like quantum states. So what this state happens, it forms a resonance in the cross section. So that means the sudden surge in the ionization. Not only that, these resonances also show up as absorption line in the astrophysical spectra if the lines are strong and well separated. So how do they look like and where we expect to see them? So photoionization is happening everywhere, wherever there is a light source. For example, this shows the picture of Lambda Centauri, which is a plasma nebula. In this nebula, there are a lot of stars, so the source of photons. So they are ionizing the plasma. So these are like photoionized plasma. The photoionization is happening all the time there. So we can have absorption line. Also in our own sun, we also have a photosphere, this thin atmosphere outside it, where also the photoionization is happening all the time. So how do they look like? So this is like photoionization spectrum of phosphorus 2 or single ionized phosphorus. Measured, this spectrum was measured at the Advanced Light Source at the Berkeley Lawrence National Lab in California. So this was measured by my colleague, or also collaborator, Guillermo, and we can see the whole spectrum, but we cannot identify the whole thing until we do the theory, whether all these lines are real or there's noise or there's only one state of phosphorus or other state of phosphorus to resolve all of those. So we need to do the theoretical calculations. So here, so the top panel shows the same spectrum, but enhanced, and the bottom one shows the calculated cross sections of phosphorus. In this calculated cross section, I have calculated many of them, but it seems like that only the lowest 5 states of phosphorus 2 make up most of the features in this observed in the measurement. So you can see that this feature is forming largely from the levels of the ground state. So these are the 3 levels belonging to ground state, triplet P even state. So you can see this feature. And this one is forming from one, just a low-lying excited state, which is singly D state. And these this weaker features also coming from another state called quintet S odd states. So theoretically, all these states can be calculated because they follow the quantum states, but experimentally, it is usually a combination of all states and sometimes other effects that influence the experiment, like a field effect or beam spread or the laser beam spread all together. It can make only or most of the spectrum, but not the 100% of the predicted spectrum. So this is photoionization. So next, another process, atomic process called electron-ion recombination, which also involves a photon which can be detected as a recombination line. So let's say this is, for example, this is an ion, and an electron is moving and finds there is a vacancy. So it decides to join the ion. So when you join it, it has to shed out its energies, which comes out as a photon. And that photon is detected as a recombination line in the spectrum. So photoionization and recombination, they are actually inverse process. Even in the absence of photoionization, recombination can take place with emission of photon. So this shows the ion and electron, they are combining together and giving out one photon. Again, like in photoionization, between the initial and final state, a doubly excited state can form based on the total energy in the process. See, doubly excited state is also the autoionizing state. It also introduces resonance in the recombination process or recombination cross-section. So how do they look like, and where they are seen? Just like photoionization, recombination can not for photoionization. Recombination take place when you have photoionized plasma, like this is a Crab nebula. So we have a light source, the photons are there, so it is photoionizing. Also we are having electron ion recombination, and so having some ionization balance in the system. So in this plasma, both processes are happening. But even in the place where there is no light source, so these are the galaxies, but in between galaxies is empty space. In this empty space, we still have elements moving, ions moving, electrons moving, and when they come close together, they can recombine. And even though there is no photon source, recombination can take place. So the way we have studied recombination, electron recombination, is through a unified method that my colleague Anil Pradhan and myself developed. It takes care of the two way of recombination. When we call it a direct recombination, like electron combining, and when we have an intermediate state form, we call it dielectronic recombination. So both radiative recombination and dielectronic recombination are happening in nature, and they are inseparable. So how do they look like, recombination process? This one showing electron ion recombination of phosphorus 2. Again, you can see that, with energy, photoelectron energy, these cross sections are having all resonances. These are the resonances, and these red arrows showing various excited states of the core ion. That means phosphorus 3. What happens, whenever there is a recombination, usually it enhances and goes up to that, then it goes down. So again, resonances, this is kind of nature of recombination. Again, it peaks and then it goes down. But this is in scale, log scale. So you cannot see the whole thing, but it can be seen here in this lower panel, where it is similar to this, but it is the recombination rate where it has been multiplied by the electron velocity. You can see that, they peak, then they go down. They peak, recombination peak, then it goes down beyond it. So that kind of the feature of recombination. This plotting of this spectrum is to provide the spectrum for experimentalists, because this feature can be measured in the laboratory. Okay. So this one shows the feature of recombination with temperature. So electron can combine in the ground state of the ion, or it can combine to various excited states. Ground state feature is pretty smooth. Recombination is high at low temperature, but as you go down with temperature, it goes down. So, and the low-lying levels show similar feature, but as we start to go higher levels, then we can see, various kind of features. So it goes down, recombination, but as the temperature goes up, the recombination again rises. You can see here too. These are because of dielectronic recombination. Even though radiative recombination dominates at low temperature, dielectronic kind of recombination start to dominate in the higher temperature and peak, like this kind of bump, right? So what happens? We combine all this recombination together, all excited, so to get the total recombination rate for that ion, because that rate is very important to calculate the ionization fractions, either in photoionized plasma or also in coronal plasma, where there is no photon source, but electron collision are ionizing the elements or ions. So this shows a total electron ion recombination of phosphorus. So again, we see at low temperature, recombination is high, it is going down with temperature. And at this point where the ion is most probably exist in various in the plasma because of the temperature, it is the point where radiative recombination and dielectronic recombination both interfere. And this is the point which should be treated very accurately, because this is where they are abundant, and it can be treated very precisely with unified method that we developed. But as the temperature goes up, then dielectronic recombination starts to dominate and forms a peak, and with higher temperature, then recombination goes down. So these are the characteristic features that are needed to calculate the ionization fractions at different temperature of the plasma. Okay, so now I'll talk about the fourth process, which also involves photon, which is electron impact excitation. Same electrons are moving in space and collides with this atom or ion here, and then excites and goes away. That means part of the energy has been transferred for the excitation, but this excitation is short-lived, so electron will go down, it will give out a photon, which will be caught by an observatory or telescope. So you can see that electron combining, combining with the ion, and not combining, exciting the ion, and ultimately which gives out a photon. So this photon carries all the information about the process as well as the electron, the energy of the electron. The quantity that we calculate for this process is called the collision strength. Okay, so it is related to the cross section, right? So where this kind of photons are important, this is just one example of this photon created through EIE, or electron impact excitation, is very important. This process is happening everywhere. This is the most common process in astrophysical spectra or in space. Okay, so this is an example. This is a galaxy. It's called ultra luminous infrared galaxy. These galaxies have one thing, that it is also like a birthplace of stars. So many stars are being born. The temperature is not too hot. It is hot enough, but there are a lot of dust around. So the photons being created are also get absorbed by the dust around it. But the photons created by EIE for low excitations are some photons which are not very popular. They don't get absorbed by the dust, so they can escape through this dust and travel in the space, and we can detect them. Once we detect them, we can get all kinds of information. So this process is also very important. So it can happen also on phosphorus. So I'm showing the example. that we did calculation on phosphorus 3. So 2 electrons knocked out. So that's phosphorus 3. This shows the collision strength of phosphorus 3 of the lowest excitation. Again, also in the collision strength, we have these quantum states, Rydberg states, which introduces this kind of resonances in the collision strength. And it is very important to include all the resonances in the calculations. And the way to include all these resonances is through the R matrix close coupling approximation. It is the most powerful method to calculate any atomic parameter for number of atomic processes. So R matrix has been used to calculate this collision strength. Once we know this collision strength and their features, then we can predict accurately various quantities, the intensities and various diagnostics of the plasma from where it is coming, the photon is coming from. So once we know, we know the collision strength, what we do, we take the Maxwellian average with temperature of the plasma, and that quantity is called the, we usually call it Maxwellian average collision strength, and denoted by Q. And Q is related to the intensity of the light we see. Also, it is related to the density of the ion in that plasma, so which gives us the diagnostics. So it shows the effective collision strength, the average collision strength with temperature of this excitation to the lowest state, which gives now a photon of 17.9 micron. So this transition that we saw the characteristic and averaging out, we get these kind of features. It tells us that the excitation increases with temperature, and it becomes 3 times more around this temperature, and it shows a broad feature. And not only that, this temperature corresponds to the planetary nebulae plasma. Planetary nebulae is the dying star. So we can easily see if the phosphorus is there, this kind of feature should be easily seen. Okay, so this is the way we study electron impact excitation. So these are the 4 processes I mentioned to study. And so we have been carrying out systematically to see the features, to try to get the more accurate underlying signs so that they can be used to analyze the observed spectra and also predict the spectrum where to look for. So all this work also done under 2 projects, we can say. One is the opacity project, the other one is the iron project. Those projects started long ago in 1983. Paper started to come in 1987, and objective, it started to explain the Cepheid variable, but objective is to solve various astronomical problems. So, all right. So we belong to this to international collaboration, and our objective is to study in detail all the atomic processes accurately and solve, any underlying, study the underlying science, solve many astronomical problems. And we found new physics in photoionization. We developed this new method, unified method, and we produce a huge amount of data too for atomic processes. And those data are available in 2 databases. One is called the top base at CDS France. Tip base is also at CDS France. OP server, that is for monochromatic opacities, available at Ohio Supercomputer Center. And NORAD atomic data that I maintain, it is at the astronomy department of the Ohio State University. My last slide shows the book that we have written, The Atomic Astrophysics and Spectroscopy. This book explains the underlying science of the things going on in the space and how to study them through spectroscopy. It shows the link between the atomic physics and astronomy. It's the bridge between the two. So this is all I like to present today, and thank you for listening.
Overview of Sol-Gel Advanced Nanostructure Materials Application by Professor Ines Batisha, National Research Center, Egypt. Our aim from this work is three important applications: photonic, information storage, and biosensor application. For photonic application, we prepared the silica gel, the phosphate by melt quenching technique, and silica titania, and hybrid phosphosilicate polyamido-amine hyperbranched polymer, all of them doped with rare earths and prepared by Sol-Gel technique. Also, the barium titanate and barium tin rare earths was prepared for information storage application, and zinc oxide nanorod drawing on the surface of plastic, silicon, silver, and many different kinds of substrate in order to act as a biosensor used for measurement of ion concentration like iron, calcium, and glucose was prepared for biosensor application, and many characterization were done for these materials. The technique used in this in the preparation of this materials was the Sol-Gel technique available in our Sol-Gel lab in the NRC, and through a special unit of electric and dielectric measurement of material. It have four way of preparation: spin and deep coater for thin film, and monolith technique and powder technique. This is the spin coater scheme of for the preparation of thin film, and here is the equipment of deep coater for preparing thin film by deep coating technique, and the monolith, which is mainly depend on the aging the material after choosing well the precursor and mixing it and put it in boxes, plastic or glass boxes in a drying oven for 21 days at 60 degrees centigrade. Many advantages were obtained from the Sol-Gel technique. The most important one is that it produces at the end immediately a product in a nano scale. Many matrices was prepared, such as silica gel, barium titanate, silica titania, silica phosphate, phosphate by melting. All the past were by Sol-Gel technique, and we did some phosphate, prepared some phosphate by glass by melting, and the silica titania also. All of them are doped with rare earths and transition element for many application which we hoped to obtain it. And here we will begin with the biosensor application of using the zinc oxide nanorod, in which the zinc oxide nanorod is prepared, the thin film is prepared by two steps: the seed layer by Sol-Gel technique, and the by aqueous chemical gross deposition by aqueous chemical gross at 90 degrees centigrade. Here we have an X-ray of zinc oxide nanoparticle seed layer heated at 100, 150, and 250 degrees centigrade, and then at the end those are three seed layers steps, and number D was for the preparation and the deposition of zinc oxide nanorod by chemical aqueous chemical gross at 90 degrees centigrade, and we can see strong and sharp diffraction peak at 002 plane, demonstrating the zinc oxide nanorod grows along the 002 plane with orientation perpendicular to the c-axis direction. And here we have the field emission cross-section view of the zinc oxide nanorod annealed at 150 degrees centigrade, and we can see homogeneous and well alignment zinc oxide nanorod, and we enlarge it a small area to see the hexagonal yardite structure well defined and well prepared for this film. To study the effect of the seed layer under the zinc oxide nanorod on this zinc oxide nanorod, we prepared two films, one with 3 layers of seed layer and the other with 6 layers. We can see well that A with the 3 layer is less and smaller than the rods of the deposited on the 6 layer of the seed layer of the zinc oxide, and we focusing on the hexagonal structure in figure C by enlarging a small area to see the hexagonality, the homogeneity, and the alignment, the well alignment perpendicular to the substrate of the nanorod prepared. Here is for the zinc oxide nanorod after and before the coating it with ionophore membrane for preparing it for biosensor, zinc oxide biosensor. Here we have the silver wire with 25 millimeter in diameter, with deposited by seed layer at 250 degrees centigrade as a panoramic view up and down. We focusing with highly magnified on a small area to see the alignment and the hexagonality and the homogeneity of the rod prepared on this silver wire. The last one of this zinc oxide nanorod is to use the borosilicate glass capillary tube with 0.5 micrometer in diameter as a substrate for this zinc oxide nanorod. The nanorod was distributed as we see in the lower figure. The nanorod was rods were distributed in it's wrong here, it is not was, it were distributed uniformly in diameters range between 26 up to 38 nanometer in silver wire, and it display from we can display from the figure that the nanorod were found to be perpendicular to the substrate and well alignment. However, its diameter is in the range between 22 up to 24.2 nanometer for zinc oxide nanorod deposited on the tip of the capillary tube. We can conclude that from the figure of the zinc oxide nanorod of deposited on the capillary tube and on the wire, silver wire, they are in good in rod shapes, hexagonal cross section, and have homogeneous distribution perpendicular to the substrate with small diameter. The application using the zinc oxide nanorod biosensor for measuring different ions by using different kinds of substrate as a biosensor for ions concentration measurements like iron, calcium, and glucose in excellent solutions such as plasma and blood. For that, we prepare zinc oxide nanorod thin film covering with different kind of a specific polymeric membrane which are compatible for measurement of different kind of biological ions such as iron ionophore membrane for selective iron biosensor, calcium specific ionophore membrane for calcium biosensor, and enzyme glucose oxidase immobilization for glucose selective biosensor. And those are the publication from this field of work in by our teamwork, and our colleague Hanan Abdelzahar Abdelwahab will discuss in more detail the glucose biosensor in this interesting conference. We will move now to the waveguide application and optical amplifier. First of all, I would like to introduce to you the basic structure for the most common waveguide geometry. We have three type of waveguide: planar waveguide, channel waveguide, and optical fiber in C. In a planar waveguide, in all the three types, we have the same property or the same phenomena. If we succeeded in obtaining it, we can apply the waveguide, which is the trapped light by total internal reflection in the film. The film will be a middle media in the planar waveguide is in the middle of the two media. In the channel waveguide is a groove inside this substrate, and at the end for optical fiber is it's surrounded. The film is in the core and is surrounded by a cladding area. Now for the same application, we choose the silica phosphate doped with erbium ion, and we found that it is transparent, but it have no time to show all the figures. We drawn the transmission, and we obtained 92% of transparency, which is enough for obtaining waveguide to support waveguide film for waveguide application and confinement of light. And this sample was studied by field emission scanning electron microscope, and we found it have a bigger thickness than 1.5 micrometer up to 1.8 micrometer at constant and its refractive index at constant wavelength 1450 nanometer equal to 1.7 after centering this planar waveguide thin film layer system at 500 degrees centigrade. The obtained data confirms the possibility of this waveguide activated by erbium ion to be successfully prepared and allowing the low loss systems waveguide fabrication, which is the goal of our work. And we follow it by studying the transmission, a high resolution transmission electron microscope for the three concentration prepared, which is 1 erbium, 2.5 and 3.5. All the three concentration was centered at 6500 and 900 degrees centigrade. All the sample at A, D, and G, the sample centered at 60 degrees centigrade were found to be at amorphous phase by increasing the centering temperature from 60 degree to 500 and 900 degrees centigrade. The crystalline phase appear and nanostructure appeared and we obtained a spherical shape of the particle, confirming the presence of nanophase, and no clustering appear due to the presence of phosphate inside the matrix. We studied the room temperature near IR photoluminescence spectra for these samples. The sample doped with 3.51 up to 3.5 mole percent of erbium. Two concentration, we choose two concentrations, the 1% and the 3.5. We found that the 3.5 increased in intensity by studying the photoluminescence, and the transition from the first excited state 4I13/2 to the ground state 4I15/2 occurs at the wavelength of approximately 1.1500 nanometer, which is our goal from this work. Two, by studying the effect of increasing the centering temperature from room temperatures than 100 up to 950 degrees centigrade, we obtained a decrease in the intensity of the peak we obtained a quenching mechanism by at lower temperature, and an increase in intensity by increasing the centering temperature. The quenching technique is normally obtained because the presence of the OH group at lower temperature, which causes this quenching technique. And here we have the laser beam passing through our film prepared at 0.5 mole percent of erbium. We doped here the silica phosphate by 0.5 mole percent of erbium, and this is the way of the laser beam excited material by argon laser at 514 nanometer. Now we will move to other samples for our important application of optical amplifier and laser. We prepared the phosphate by melt quenching technique, and we obtained an amorphous phase, two sets of the samples, phosphate doped with erbium and barium and potassium. The first set of samples are of erbium doped instead of barium, and the second is doped, the erbium ion is doped instead of potassium, and we obtained the amorphous phase for all the prepared samples. And the second figure is for the absorption coefficient of this material, and the absorption obtained bands are assigned by comparison with the energy levels of previously reported erbium glass system. No change. We succeeded in preparing then our samples. The absorption coefficient increases by increasing the erbium ion concentration. It is worthy to note that the linear increase of the absorption with the erbium content indicates the absence of rare earth chemical clustering due to the presence due to the homogeneity of the host material of phosphate with some doping such as barium and potassium. The emission around, we have at the third figure here, we can see the photoluminescence, which is succeeded well, and we obtained emission around 1536 nanometer, attributed to the transition from the 4I13/2 up to 4I15/2. Other things we prepared and calculated and drawn the gain spectra as a function of wavelengths at different values of population inversion rates. P equal 0.2, 4, 0.6, 0.8, and 1 for the two sets instead of barium and instead of potassium. The gain was calculated by the equation in front of you, and then the gain spectra as a function of P, the population, the population inversion as a function of the population inversion of the two sets of samples. The figure has maximum inform or show the maximum value and peak position versus P for the prepared samples. It can be noticed that the amplification would be achieved around 1594 nanometer for low value of P at 0.2, a gain of 0.2 decibel centimeter minus 1, and nearly flat band widths are obtained. When increasing P, the gain value increased and the peak shifts to the shorter wavelengths. For total population inversion P equal 1, the gain peak centered around 1522 nanometer for all the samples. By increasing the erbium content, the gain increased. It can be noticed that the gain is slightly higher for samples doped with erbium instead of B than the sample doped with erbium instead of K. And this is the level energy, the internal energy level diagram of erbium and ytterbium together. And here we have the expected diagram of the total internal reflection process, light propagation inside the silica phosphate doped with erbium from 1 up to 3.5 mole %. And here we have some publication from the quenching, melt quenching phosphate doped with erbium and activated with ytterbium, sometime with ytterbium at 2019 and 2020. And now we will move to other third publication, which is the barium titanate doped with some rare earths pure and doped with some rare earths. First of all, we prepared it as pure sample, and we studied the dielectric in wide range frequencies between 1 and 1 megahertz and 1 gigahertz. We studied our drawn the permittivity, epsilon, dielectric loss, and the conductivity by using the K-site E4991B, which is in our lab and in our special unit of electric and dielectric measurement. We studied the transmission electron microscope of the barium titanate, which give us homogeneous and no accumulation appeared, and informed us about a nanostructure and the diameter of particle, which is begin from 18 up to 36 no more. Then the sample is in nanoscale, and here is the sample, the transmission electron microscope of the sample doped with tin oxide, and here we have the tin oxide, the barium titanate doped with tin oxide and doped with erbium ion, and doped with erbium and ytterbium ion. The transmission electron microscope shows that the sample are in nanoscale, and here is the morphology detected by the on the right by the scanning high field emission scanning electron microscope. Here is the steps of preparation of the barium titanate in the solution case, then by heating it and began in gel case, and after that it will be, here we are in 350 degrees centigrade as drying, and after that we will centering it up to 1050 degrees centigrade to obtain single phase of barium titanate and barium titanate doped with SN. It will give us a very clear powder. Here we have the X-ray diffraction pattern of the pure sample beginning from 350 degrees centigrade up to 1550 degrees centigrade. And here is by increasing the concentration of the tin oxide, and we found that the tin oxide is the best way, or the best condition, the optimum condition to use it for doping it with rare earths is the tin doped at 10 mole %. those many publication was done for this 33 international publication, but I will introduce here the most recent one only, which is for publication in 19 and 20 in 2020. Now we have the silica titania we obtained. Here we have the silica titania in monolith, pure in monolith form, and then heated at 500 degrees centigrade. And here B, C, and D is by doping it with 0.4 holmium ions and 1 mole holmium ion and 1.4 holmium ion at 1000 degrees centigrade. In this case, it's a brochite. We have anatase phase only at 500 degrees by centering it at 500 degrees centigrade, and at 1000 the brochite and the rutile appeared. And also we obtained a high transparency film of silica titania, which can be used well as waveguide in waveguide application. And we studied the upconversion emission variation of holmium ion concentration with the power and with the concentration of holmium ion. And we have here final conclusions that the aim of the present work was to develop the phosphate phosphosilicate and hybrid phosphosilicate polyamido-amine hyperbranched polymer erbium nanocomposite co-activated with ytterbium ion as sensitizer toward planar waveguide and amplifier application. The zinc oxide nanorod act as a biosensor used for measurement of extra intracellular ions. The overall aim of the work is in biosensor field is to help the society by introducing a new advanced biosensor technique to help in early diagnosis of some common disease such as diabetes and cancer by using intracellular ions measurement in the very near future, inshallah. For information storage, we prepared the barium 10 and barium 10 rare earths. Thank you very much.
By the end of the first session of the second day of our conference, we would like to thank all the speakers for their very important and nice talk. And now we start our open discussion. Thank you.